Hi everybody, this is the second chapter in the basic Immunology Lab Techniques. By the end of this chapter, the student is expected to be able to state the different conditions for precipitation to occur. Last time we studied agglutination. This time we are studying many other antigen-antibody reactions, one of which is precipitation. You should be able to list different conditions for precipitation to occur, list different examples of precipitation, interpret the results of the provided precipitation tests, explain the principle of neutralization tests. You should also be able to explain the principle of enzyme-linked immunosorbent assay and immunofluorescence tests. Precipitation describes the reaction between soluble antigen and antibody when both meet at a zone of equilibrium. In this case, a visible precipitation reaction occurs. Agglutination and precipitation are maximized when multiple antibody molecules share the binding of multiple antigenic determinants, a condition known as equivalence. In vivo, the precipitation of such complexes from the blood is critical to the trapping of pathogens and the initiation of the immune response in the secondary lymphoid organs, as well as the initiation of the pathologic phase of many immune complex-mediated diseases. In vitro, the kinetics of such reactions can be observed by titration or gradual measurement of antigen against its specific antibody. Precipitation reactions can be done in a variety of ways. We can have precipitation in solution, which is known as the ring test, or precipitation in gel. We use a certain gel called agarose gel, which can be performed in the form of single radial immunodiffusion, double diffusion, or immunoelectrophoresis. And we will be discussing each one of those in more detail. Now, if we start with precipitation in solution, this is performed by the ring test. Basically, the serum, which potentially contains antibodies, is introduced into a small diameter test tube, and the antigen is then carefully added to form a distinct upper layer. After an incubation period of up to 4 hours, a ring of precipitation or a ring of precipitate forms at the point of contact or the interface in the presence of the antigen-antibody reaction, in the presence, that is, of a positive case. This test is used, for example, in the diagnosis of infectious diseases by detection of the microbial antigens or antibodies in a patient. Now, if we take the gel precipitation, now the first mechanism of performing precipitation reactions in agarose gel is the double diffusion method. Now here, as the name implies, the soluble antigen and its corresponding antibody are placed in two separate wells in a plate of gel. The reactants will diffuse and form a concentration gradient. Optimal proportion is reached at some point between them, and antigen-antibody complexes form a white line or a precipitine line. The main application of the double diffusion precipitation technique is ELIX test. Now, this is an in vitro test which is used for the diagnosis of toxin secretion by strains of Corynebacterium diphtheriae. Now, you must have heard of Corynebacterium diphtheriae. This is a particular bacterium that causes childhood illness called diphtheria. The most important issue is not the bacteria itself, but some strains of it which produce the toxin. So it's really important for us to test if the strain of diphtheria isolated from a patient is actually toxin-producing or non-toxin-producing. And this is the way we do it. In this case, a sterile filter paper is impregnated with the diphtheria antitoxin, the antibody to diphtheria, and is embedded in the serum agar culture medium. Isolates of Corynebacterium diphtheriae, the organism, from the throat of a case or carrier are then streaked across the plate at right angles to the antitoxin strip. If the organism is toxigenic, that is to say it produces the toxin, then the toxin will diffuse from the area of growth. The antitoxin, which is the antibody to that toxin, will diffuse from the filter paper. The toxin and antitoxin will meet at optimum concentration, and precipitine lines again are formed. And this usually happens within about 18 to 48 hours. This is what the test looks like when a toxin-producing strain is tested. As for single radial immunodiffusion, it is used, for example, for the quantitative estimation of antigens, for example, serum protein concentrations. If you want to test the presence and the number or the amount of serum proteins, we consider them the antigens, and this is how we do it. Also for various complement components, various immunoglobulin isotypes, and so on. This method is carried by incorporation of the antiserum, or the antibody against the tested antigen, in the agar solution during agar preparation. Again, agar is just a gel that is prepared in plates and is suitable for the growth of many organisms. Thus, the antiserum is uniformly going to be distributed throughout the agar gel. A ring forms where the antigen and antibody meet at the zone of optimum concentration. The greater the initial concentration of antigen in the well, the greater the diameter of the precipitation line. By running a range of known antigen concentrations on the gel and by measuring the diameters of precipitine rings, a standard curve can be constructed, as you can see here. The antigen concentrations of unknown samples, or the patient's serum, are run on the same gel and can be calculated from the standard curve. Let's take a closer look at how this is done. Basically, the application of radial gel immunodiffusion, again, is quantitation of the estimate of certain antigens such as serum proteins. In this case, wells are filled with agar gel impregnated with fixed amounts of antibody to the antigen in question. Standard pre-prepared antigen concentrations are added to the first few wells, and the last two wells in the row have the two unknown serum samples. That is, the samples with unknown concentrations of the specific antigen that you're looking for. You allow time for diffusion of the antigens into the wells, and precipitation lines form depending on the concentration of the antigen. You can see that the first 4 wells demonstrate increasing ring diameter as the antigen concentration is increased. A curve is then prepared, correlating the ring sizes with the concentration of the tested antigens. And the size of precipitation rings formed around the tested samples can also be measured, placed on the curve, and the resulting antigen concentration is read off of the curve. Next, we move on to neutralization. Neutralization is an antigen-antibody reaction in which the harmful effects of a bacterial toxin or virus are actually blocked by specific antibodies. That is, the antibodies neutralize the effects that the virus normally has on the cells. Now, one of the uses of this technique is in the diagnosis of certain viral infections, and this is actually the basis underlying the virus neutralization test. The presence of antibodies against the virus can be detected by the antibody's ability to prevent cytopathic effects of the virus in cell culture. Thus, if a virus normally attaches to a cell and causes cell lysis, the presence of antibodies in the serum of the patient can be detected by the inhibition of this cell lysis in the presence of the patient's serum, which supposedly contains the antibodies. This is also the basis of the hemagglutination inhibition test. In this test, the effect of a virus is to cause agglutination of RBCs, then the presence of antibodies in the serum of a patient can be detected in vitro by the ability of the serum to neutralize the virus, and so no heme agglutination will occur. ELISA is a biochemical technique which is used mainly in immunology to detect the presence of an antibody or an antigen in a sample. The indicator system in ELISA is an enzyme attached to an antibody and a substrate. The enzyme converts a colorless substrate, which is known as the chromogen, to a colored product, indicating the presence of antigen-antibody binding. In other words, this is an antigen-antibody reaction that is detected by the production of a colored substrate that is produced due to the action of an enzyme, which would only be present if the antigen or the antibody in question are present. The procedure can be direct to detect antigen or indirect to detect antibody. In direct ELISA, antibodies specific for the antigen in question is captured to wells of a plastic microtiter tray. The specimen, which may or may not contain, of course, the antigen, is added and incubated with fixed antibodies in the plate. Washing is done to remove any unbound reaction so that only specific antigen-antibody reactions that is fixed to the plate persists. We then add the specific antibody that is labeled with an enzyme. In positive cases, the labeled antibody becomes attached to the antigen, and this is called the sandwich technique. Addition of a substrate to the wells allows the antigen detection by a colorimetric reaction due to the effect of the enzyme on the substrate. In negative cases, enzymes labeled antibodies will not find an antigen to be attached to, and it will be washed away. So no color change will be detected after addition of the substrate. In indirect ELISA, which is basically used for antibody detection, the antigen specific for the antibodies in question is adsorbed to the inner surface of the wells of the microtiter plate. Serum, again, which may or may not contain the antibodies to that antigen, is added to the antigen fixed in the microtiter plate. Again, in positive cases, the presence of antibodies bound to the antigen can be detected by the use of anti-immunoglobulin or antibody to the antibody labeled with an enzyme. On addition of a substrate, the color change will be detected. In negative cases, no antibodies are bound to the antigens, and therefore anti-immunoglobulin labeled with an enzyme will be washed away, and no color changes will be detected after addition of the substrate. This is usually done in multi-well microtiter plates so that different dilutions of the serum can be easily prepared. Let's take a closer look at one of those wells. To perform the assay, the wells are coated with the antigen of interest, which is commercially prepared by the manufacturer of the plates. The wells are filled with the different dilutions of the patient's serum, which supposedly contain the antibodies to the antigen, if, of course, the patient is diseased with that antigen. If antibodies to the antigen are present in the serum, they will bind to the antigen fixed in the bottom of the wells. The patient's serum may contain many different types of antibodies, but only those specific to the antigen in question will stick to the wells. Non-specific antibodies will be washed. Next, we add antibody to human antibodies, and this is an animal antibody against human antibodies, and is covalently linked to an enzyme. When the substrate specific for that enzyme is added, a color reaction occurs. Thus, if a color change happens in the well, this indicates that the substrate found its enzyme, which means that the enzyme stuck to an anti-human immunoglobulin antibody, which has detected a human antibody that got bound to the antigen in the plate. Remember, the source of that latter antibody was the serum of the patient, so this indicates that the patient does indeed have specific antibodies to the antigen in question, which also means that the patient has been exposed to the antigen. The final antigen-antibody detection system that we will be discussing in this lecture is immunofluorescence. Here the principle is that a fluorescent substance such as rhodamine B can be covalently attached to an antibody molecule and made visible by fluorescent microscope with UV light. Thus, the presence of an antigen in a sample can be detected by adding a pre-prepared antibody against that antigen, labeled with a fluorescent dye, and detection of fluorescence indicates the attachment of the antibody to that antigen, thus indicating the presence of the antigen, which would otherwise not be detectable. Immunofluorescence can be used in detection of antigen or antibody, in which it is termed direct or indirect immunofluorescence respectively. Immunofluorescence is also the basis of flow cytometry, a technique used in modern era research for cell labeling and counting. And we will be discussing this in the next lecture. In primary or direct immunofluorescence, a single antibody is chemically labeled with an immunofluorescent dye. This antibody recognizes the target antigen and binds to it. The dye can be detected by ultraviolet microscope. It is used to detect antigens. In indirect immunofluorescence, two antibodies are used. The first, or the primary antibody, recognizes the target molecule and binds to it. The second, or the secondary antibody, which carries the immunofluorescence dye, recognizes the primary antibody and binds to it. This is the end of the chapter, and you can now move on to doing your quiz. Thank you.